who has joined us through the link, please proceed to fill in the attendance form that we have put in the chat box. Secondly, please note that we will only issue it to participants who complete the attendance form. Thirdly, we will put the participant on mute during the webinar to minimize the noise disturbance. If you have any questions or need any assistance, please use the raise hand button. Our staff members will attend to you as fast as possible. Finally, for the question and answer session, please post or write your questions in the chat box as well. We will also have a group photo at the end of the webinar. Thank you and have a nice session ahead. Assalamualaikum and a very good afternoon. Selamat datang and welcome to another series of our UiTM Food Technology webinar hosted by the Faculty of Applied Sciences University Technology Mara, UiTM, Malaysia. Before we proceed further with our event today, please allow me to invite myself to the stage for a short prayer to bless our event today. Bismillahirrahmanirrahim. Alhamdulillahi Rabbil Alamin. Wassalatu wassalamu ala ashrafil anbiya'i wal mursalin wa ala alihi wa sahbihi ajma'in. Allahumma innaka khalaqul azim, innaka sami'un alim, innaka ghafurur rahim, innaka rabbul arshil azim, innaka barrul jawadul karim. Ya Allah, ya Tuhanku, Engkaulah yang mempunyai segala kepujian, Engkaulah yang berhak menerima segala kepujian, Engkaulah yang berhak menerima segala kesyukuran, Engkaulah yang memiliki segala pemerintahan, di tangan Engkau segala kebajikan, kepada Engkaulah kembali segala urusan. Allahumma ya Karim ya Rahim, cucurilah rahmat dan rahimmu ke atas majlis UiTM Food Technology Webinar yang diadakan pada hari ini. Berkatilah ia dari awal hingga akhirnya. Ya fa'alul lima yurid, kami juga memohon perlindungan daripadamu dari segala perkara yang boleh mencacat celakan majlis kami dan daripada segala perkara yang melalaikan kami dari berbuat taat kepadamu. Kepadamu jua kami serah segala urusan kami. Rabbana alaika tawakkalna wa ilaika anabna wa ilaikal masir. Rabbana atina fid dunya hasanah wa fil akhirati hasanah wa qina azabannar walhamdulillahi rabbil alamin. Thank you. Ladies and gentlemen, our webinars today topic shall be on methods to determine antioxidant capacity in food samples, presented to us by a specially invited speaker from University Putra Malaysia, UiTM, Professor Dr. Amin bin Ismail. Dr. Amin bin Ismail is a professor in the field of chemistry and biochemistry of food and analysis of food composition at the Department of Nutrition, Faculty of Medicine and Health Sciences, University Putra Malaysia, UiTM. He is currently the Director of Center for Quality Assurance, CQA, in UiTM. His research areas focus on exploring of health-promoting compounds of underutilized plants for obesity prevention, human health, and food composition database. Dr. Amin has graduated more than 15 PhD and 25 master's research students. To date, he has more than 250 publications, and his H index is 43, based on Scopus. Nowadays, Dr. Amin is actively serving as an editor or an editorial board member at several national and international journals. Since 2012, he has been appointed as a visiting researcher at the Guangxi Academy of Agricultural Sciences, Nanning, China. Only recently in 2018, Dr. Amin had also been proudly presented with the Distinguished Malaysia's Research Star Award for a high impact paper in the Agricultural Sciences category. Therefore, without further ado, I pass on this ceremony to our moderator, Professor Dr. Zaibunnisa Abdul Haiyee, to kickstart our webinar. The floor is yours. Thank you.
Thank you, Mr. Emcee, Mohammad Ikrami bin Salehuddin. Welcome to this webinar. Assalamualaikum warahmatullahi wabarakatuh and very good afternoon to everyone. Welcome Prof. Amin, Ismail, all the way from UiTM with us. Okay, thank you. Thank you, Prof. Today we're going to have interesting webinar titled Methods to Determine Antioxidant Capacity in Food Samples, which is a very interesting topic and a lot of people here out there really want to know what are the current methods that people use. Especially this seminar or webinar is actually we invite Prof. Amin to share his knowledge and expertise with us for our master student, food science and technology student. Is part of the topic that should be delivered to students. So when we look at the topic, there's a lot of demand and people ask to join. So that's why we open the webinar to everyone, so everyone can benefit from this sharing session, which is I think it will really helpful to all the food scientists. I've seen some from even international participants are already here with us, Prof. Amin. Okay, so let me just a little bit introduction about Prof. Amin Ismail itself. Prof. Amin is actually a very renowned food scientist, originally from food science and technology. Now is already in the Faculty of Medic. Is actually renowned Prof. Amin for so long, back from when we were studying together. So Alhamdulillah, he's here to share his expertise with us. And I asked Prof. Amin, can you please tell me how many invited presentation that you have actually joined so that I can highlight in the webinar? He says too many because he has so many years of experience in this field. I remember when he was doing his PhD, already started doing antioxidant activity analysis for his PhD. So very vast knowledge he has in this field. I just pick up some international lecture that he already given a topic that I noticed, titled Bioactive and Bioactivities of Underutilized Tropical Fruit in Second Food Chemistry Conference at Spain last year. Yes, Prof. Amin. So it's a very common topic for Prof. Amin, but of course for us, we want to know from the expert how actually his point of view on this topic. So antioxidant activity is a really important analysis for food science and technologies, especially during now the COVID era, whereby you have to consume a lot of nutritious food, which can actually give you more, boost your immune system and all that. So it is for us to know what is the best method or what is the appropriate method that we can actually use to determine the antioxidant capacity of the samples. So with that, without further ado, I would like to invite Professor Dr. Amin Ismail to proceed with this interesting topic for today. The floor is yours, Prof. Amin. Thank you.
Thank you. Listen. Are you able to listen the voice? Is it clear?
Yes. Yes.
Yes, Doctor.
Yeah, because I think that Zaibunnisa, I think your voice, sometimes breaking, in the between. I'm worried, also the same thing, happened to my presentation. I hope you are able to listen clearly. Okay, clear, yeah? Okay, let me share my slide. Okay, my slide, my slide presentation.
Yes, Doctor, very clearly.
Yeah. Okay, thank you so much. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. salam sejahtera. Thank you, Saudara Ikrami, for your kind introduction. My respectful Associate Professor, Dr. Zaibunnisa, like my adik, always call me Abang Amin, and then also I think the student, especially the program, under the coursework program, from the UiTM. Also I think my colleague also here, I have also my student, my ex-student, also from others university, for beside UiTM, also the international participant. I think welcome to this, the talk, on method to determine antioxidant capacity in food sample. I'm not, in UPM now. I'm from my house in Puchong. Okay, so I would say today, Saturday, this I think most of you will spend some time with family, the time with the family. I would say, Santai Sabtu, because I think we will make it our presentation, our topic more santai, more relaxed, don't tense, because don't make it this topic a heavy topic, just like something you are just sharing, and then you at home, so you can have a free have your coffee, and then your snack. Okay, let me begin, method to determine antioxidant capacity in food. I think okay, outline of the presentation. I would like to share, I would like to cover antioxidant history in Malaysia, that I did the information for quite some time, and then antioxidant study in Malaysia, and then a very basic principle of antioxidants, and then antioxidant capacity or antioxidant activity, because sometimes people might try to exchange between the capacity and activity. I try to ask the expert, the guru in this the chemistry from Canada, and then he asked me that no problem whether you use antioxidant capacity or antioxidant activity. I would like to look at on the chemical and biological in vitro assay, and then antioxidant capacity or activity, about the mechanism. And then I think last 2 years, there are many criticism about the antioxidant capacity or activity based on in vitro system. And then about case study on antioxidant, from where we start, from the basic, using the in vitro assay, to the human interventional study, and then I will try to make a conclusion from this presentation. Okay, history about antioxidant. I think nowadays, I think all the participants have been working or have been dealing with the antioxidant for quite some time. But I have been working with this antioxidant since 2000. But now I'm moving toward more on the human intervention study. And then look at the large diversity of analytical method, for determination of the antioxidants capacity or activity is variable. This assay differ, from each other in terms of the reaction mechanism, in terms of the oxidant or target, or probe specificity, the species, reaction condition, the temperature, the pH, sometimes also will influence the result when you prepare a working solution at a different pH. And then also how do you express your result? Sometimes the published article expressed differently. And then when you would like to compare with your own result, will be difficult for you to compare, side by side, because normally in terms of the unit they use, maybe the condition, the temperature, all those, because when working with the antioxidant capacity or activity, there are many things, many variable you have to be considered. Okay, antioxidant, I think this one just like a reminder, for us also, the refresher. Antioxidant can be broadly, hold on. Okay, can be broadly defined, as any substance, that when present at low concentration compared to those an oxidized substrate, because there is the oxidation product, and then significantly prevent or delay any oxidation of that substrate. Because this the antioxidant, I believe all of you already known or already understood what the antioxidant about. Okay, the early use of antioxidant begin in the 19th century, when industrialization was becoming popular. I think the corrosion of metal, because previously they are using the oil, they have to use the oil to avoid this corrosion on the metal. So because this one of the antioxidant activity happen because due to the presence of the antioxidant. And then in the food industry, it started to use antioxidant for prevention of acidity in fatty food, in butter, cheese, oil, because I think in Malaysia also, we started using the BHT, BHA, TBHQ, because these antioxidant agent, the food additive, because most of you have a basic knowledge, information on this antioxidant. This is the static, the synthetic antioxidant, normally from the petroleum-based antioxidant. And then the application of antioxidant from food sources to therapeutic aspect started, because spices, and then we had started with the spices for the natural antioxidant. And then now, today, antioxidant has become an increasingly popular topic in most areas of scientific research. Because when now we are talking about antioxidant, most of us very familiar what the antioxidant all about. Okay, also in Malaysia, I think we are blessed, with diversity. We have a huge diversity of plant, vegetable, fruits. Not only in Malaysia, also in Southeast Asia, in Indonesia, Thailand. I think most of the tourists, they like to come over to Malaysia, because they enjoy with all the colorful fruits and vegetables. Because some of the fruit and vegetables still underutilized. That's why where we would like to do more on the antioxidant research, because this one the untapped treasure of in our country. Because when you look at we have a variety of the fruit and vegetable spices, ulam that still untapped, treasure. But this opportunity for all of you, to explore the antioxidant capacity or activity of this plant. Okay, because here my department, back 2002, now the 2022, almost 18 years, back, because we organize the scientific conference on food antioxidant, because that time still the antioxidant still new, in Malaysia. I would say in Malaysia, because we would like to have this platform for Malaysian, especially to share, to have some knowledge on the food antioxidant. During this conference, we have also the debate, the debate session, where I think will be very interesting. You can see the debate, one will be the pro, another one will be the support for that one. I think very interesting. And then in Malaysia, I think FRIM, Forest Research Institute, of Malaysia, has presented, an optimization and standardization of antioxidant assay. Because I would say FRIM, I think the first institution come up with the method development and validation of the antioxidant, because their aim to offer antioxidant testing for industries, because when you would like to offer your service to industry, you have to ensure your method you develop from your university or from your lab has gone through the method validation, method development, because when the result make sure it reliable and accurate. You cannot just simply based on, because when you're dealing with the antioxidant capacity, the assay, you might face the problem will be the inconsistent of the result. Although the same sample, but only the different time or different month, different week, you will get a different result. Because sometimes huge, because the standard deviation sometimes very high compare, because that's why you need to do some method validation of the antioxidant assay. Okay, this one I did in 2010, because I still keep this this diagram, antioxidant studies reported in Malaysia based on the Scopus. I Google, and then I found out, for example, we have it started for quite some time, since 2000, and then in 2010 you can see about 300 plus the number of articles published site under the Scopus. And then last night I tried to Google and research in the Scopus. 2015 about 500, 2018 685, 2020 1047. You can see a drastic I mean the increase in terms of the number of publication in Malaysia based on the antioxidant capacity or activity. You can see the huge people interest in this field of study. That's why if we can have understand about the shortcoming, advantages and shortcoming of this antioxidant capacity or activity will be very good for us, because now to publish not very easy compared to the previous time. Now very challenging, very challenge, because very hard last time. Nowadays, previously only one essay can be published. Nowadays, no, they have more. Sometimes you have more than one, also cannot be published. So they are asking for more, for more additional parameters. I think very challenging for us, to to work on this antioxidant research. Okay, because in every presentation, I don't leave this chocolate, because I like this chocolate, because my PhD also Dr. Zaibunnisa also master on cocoa also. I think we love about chocolate. I think sometimes we know the health benefits of the chocolate. Based on the based on the study, systematic review, or meta analysis, they found that the good, the health, the good thing about the cocoa. As working with antioxidant, we would like to know what is the compound, what is the antioxidant in cocoa that contribute to the health benefit. We cannot assume, without knowing what is the compound could contribute to this, the positive aspect. That's why come to the antioxidant research, we have to start, from the in vitro, in vivo, human intervention study, and then before we come up a sound conclusion. Okay, in 1990, I think about 10 years back, the interest in antioxidant capacity or antioxidant activity determination, the method to determine antioxidant capacity, when many new methods were developed. Previously, we have only one or two, and then nowadays, you can see we have many. We have many methods, because last week, I searched in the Google. Now, I think most of the many industry, or many industry, they develop the kit based on the assay. Previously, we use only using the spectrophotometer and the spectroscopy. I mean the method, and then nowadays they come up with the kit, that one for the commercial aspect. Because you look at there are many, and then I think sometimes very good, sometimes also not good, because what is the good thing? We have a choice, and then also the not, I mean, the non interesting thing, we have confused, which one we want to choose, because sometimes we have to choose the PPH, or we have to choose the fat. Sometimes, maybe a different paper, they the result sometimes very different, sometimes very similar, and then many things, because I think the important to have some understanding, or they call the analysis, before you choose which method you want to choose for your research. Okay, why do we interested to determine antioxidant capacity? Different antioxidants show different effectiveness in food system. Why? Due to unique molecular structure. Because quercetin, maybe they act differently compared to the catechin or epichin or anthocyanin. For example, like some of the antioxidant, maybe if you're using only single, they give a different capacity. If you combine, with this other compound, there is a different effectiveness in the food system. That's why we need to determine the antioxidant capacity in food samples. And then no current literature recognized or accepted antioxidant capacity or antioxidant activity method as a universal method. Not like protein, we are using Geldal, fat, we're using Assoc slurry. There will be the recognized method worldwide, but not for the antioxidant capacity. That's why we, very interested, to determine this in food. And then emerging research work on the health promoting properties of plant food due to their antioxidant, because we based on our old folk, or based on our parent, but when we go back to our kampung, and then some maybe our nenek, our makcik, our pakcik, because sometimes they say, oh, this plant can be, can be used, to treat diabetes, or can be used, to treat cancer, or many things. But meaning that we know maybe due to the antioxidant capacity. That's why we first, we would like to determine what is the anti capacity or antioxidant activity of that fruit before we proceed further. Because this due, to the emerging research in the health promoting properties. There are many correlation, because when you search in the literature, there are many published article or published work on this plant, able to prevent, the risk of cancer. There are many, claim can be made. For example, the industry also, they can make, like small, medium enterprise. And then also the complexity and diversity of food product, because if you're working with a single compound, should be okay, because no other factors will influence your medium, your result. When you're using an extract, because an extract, there are many compounds, because we have no idea whether the sugar, the protein, or others compound could influence your result. Because in antioxidant capacity, the assay, normally based on the colorimetry method, based on the color formation using a spectrophotometer. Because any compound could be, will be overestimate your result. There are many things you have to understand before you choose any method. You have to understand. You have to find the basic information. What is the shortcoming of that? Because not all they will give a same response. Okay, a basic principle of antioxidant. I think I have another 20 minutes. The Zaibunnisa, but about 3:20 I have to stop.
Yes, Prof.
Okay, I try to speed up. I thought, okay, a basic principle of antioxidant based on the electron, single electron or hydrogen donation to free radical. To free radical by antioxidant. This formation of complex between these free radicals and the oxidant radical, because you can look at the figure here. You can see the antioxidant. There will be the donation of electron to the unpaired electron of the free radical. This radical will stabilize. Okay, this is a very basic principle of the antioxidant. Okay, antioxidant capacity and activity. There are many methods, as I mentioned previously, because when we look at because we have ORAC, and then we have FRAP. Because FRAP, I think very familiar. FRAP, we have Kuprek. Kuprek, I think developed by one of the professor, Professor Apak from the University of Istanbul. I met him for quite some time. A very good paper published on that. And then we have ABTS, DPPH, and then TBAS. I think TBAS, this is the old method. Previously, most of the us, if we started with the TBAS, and then ferric reducing something, we have some of the one, and then LDL oxidation. And then we have also, because ORAC, FRAP, and this assay, because they are based on the quantification approach. Because for this FRAP, DPPH, and ABTS, only based on the color, formation of the color. Okay, this the in vitro the method apply in Malaysia, because when you look at there are many methods. The famous one, FRAP, DPPH, ABTS, and ORAC. Because TBAS, AC, I think normally they they would like to check, for the working with the oil sample. Okay, here a bit about the antioxidant assay, antioxidant capacity assay in Malaysia. Yeah, when you look at 1999, they start with the TBAS. Because I collected a few publication working with TBAS, using a dyne conjugation formation. And then after that, 2001, and then they are using the DPPH, DPPH, radical scavenging. And then 2002, using the linoleic acid model system. And then in 2003, they are using the enzyme antioxidant assay using XOD, and then they also they are using FTC also based on the ferric thiocyanate. And then the 2004, they are most using FAT, and then also beta carotene bleaching assay, because I started in UPM with the beta carotene bleaching assay. And then in 2006, in Malaysia also, you have expected in they are using ABTS. And then 2010, we beside using the chemical assay, we also venture, we also develop biological assay based on the inhibition of hemoglobin and LDL oxidation. And then 2011, people using ORAC, because ORAC based on this, they have a machine, and then based on the microplate reader, so they are able to determine, of a different sample. Okay, and then this I think my group publication we published in 2004. I think for quite some time, because we're using total antioxidant activity. Because that time, I'm not really vast, have a very good knowledge on the antioxidant activity. I'm always talking about total antioxidant, and then after 2004, I found out, cannot be used total, because total actually you are measure the non-enzymatic compound and enzymatic compound as a total. You can say it's the total. When you're using any method, to determine, to consider, to take into consideration the enzymatic antioxidant and the non-enzymatic antioxidant you consider as a total antioxidant. But the one we did only based on the non-enzymatic compound, because that means more on the chemical compound. So we cannot put total antioxidant activity. That's why after 2004, I'm always give advice to my colleague, to my student. You cannot use total anymore, because total not acceptable. That's why in 2006 we published using the antioxidant capacity. Always people debate about the capacity. Some of the people, they're still using antioxidant activity. And then you also can see some using the antioxidant power, antioxidant potential. But the most important thing you have to understand, whatever the capacity or activity. For me, I would prefer capacity. Why? Because activity, you are involved with the kinetic. You look at the kinetic, you can consider this the activity. Okay, this I'm not going to explain one by one all the method, just a very common one. But I think the rest, I think most of you can get the information process, like mechanism of DPPH, DPPH scavenging capacity based on the donation of the electron by the oxidant, by the antioxidant, and then you can see the starting with the color, because you can see the DPPH radical cation, there is purple, a violet color. when they will react with the antioxidant, you can see the change of the color, to the yellow. From there, you can see like estimation, what, for example, when, you have a high antioxidant, in terms of the color, you can see the changes in terms of the color. Okay, and then you look at a flat and a BTS. I think the same, the same modus operandi, because there will be the transfer of the single electron, single electron transfer. From, for example, from the ferrum, from the ferrum 3 plus, and then will be the ferrum 2 plus. You can see the change of the color, from yellowish to the blue color. Also the same for the BTS, This color you can measure based using the spectrophotometer at certain nanometer, stand absorbent, stand absorbent nanometer. Because that's why, if for example your sample has color, so might be problem. You have to look at that. For example, maybe your color like dragon fruit or watermelon. If you are, when you have your sample, but when you put it in this solution, if your color, if the color will influence your result, so you will get a high absorbance, because not due to this compound, maybe due to the color, color compound that present in your sample. Okay, all right, I think all right, based on this one, based on the rate of decay of fluorescent in the ORAC assay, because they are using I think the very light spectrophotometer, I think the machine, and then you can using this the AAPH, and then when you look at from the fluorescent, and then you can see whether they will be changed to the non-fluorescent product. Okay, this some of the result from the ORAC's assay. Antioxidant inhibit the free radical damage, because based on the rate of decay of the fluorescent in the ORAC assay, is a measure of antioxidant capacity. And then the antioxidant can be categorized as a function. You can categorize antioxidant as a function. And then, for example, here, whether there are free radical scavengers, or non-radical oxidizing agent, and then polarity, water soluble, or lipid soluble, or source of the antioxidant, this one, and then the mechanism, you have to look at also what the mechanism, whether this antioxidant as the single electron transfer, or hydrogen atom transfer, or as a chelating agent. Okay, a bit about the single electron transfer of the antioxidant method. Measure the ability of antioxidant, for example, antioxidant AH here, to transfer one electron to the reduced free radical. When you look at this, the free radical, because from the one electron here, they will transfer this one, and then the poor oxidant metal and carbonyl. This assay are solvent and pH dependent, because when you using antioxidant capacity based on this set, based on the single electron transfer, the mechanism, the solvent and pH will dependent on this your assay. And then another one, hydrogen atom transfer, measure the ability of antioxidant to quench free radical, normally the peroxide radical, by hydrogen donation. They just donation hydrogen, from this one you will get the stabilizing the radical, the free radical. Here based on the electron transfer. Because some of the method very you can consider both set and head, like DPPH and flat. you can consider they have these two, set and have. You cannot differentiate, between these two. Okay. Okay, here. Okay, here's some of the method under this single electron transfer, FLAP, DPPH, and ABTS under the single electron transfer, and then ORAC, beta carotene breaching assay based on the hydrogen atom transfer. It's the mechanism of that one. Okay, in 2015, for example, some critique on anti in vitro assay are based on the action because due to the action of fast free radical in living organism in our human body, particularly and mechanism reaction very complex. In addition, high in vitro antioxidant activity cannot be translated into prevention or treatment of NCD in human. And then, for instance, trap assay, the reaction at a low pH, 3.6. There is much critique, there are many critique about the translation of this method into effectiveness in vitro. Is it the same result you will get when you're using animal model? And then they suggest that it should be used only as a screening method. Idea of antioxidant capacity. Okay, from here, when you look at in vitro assay, whether is it chemical method or biological method, this is very important for screening purposes. Not to come up with some conclusion about any product you're working on. Okay, beside chemical assay, just now FLAP, DPPH, and then DPPH, FLAP, and then TEAC, ABTS, they are based on the chemical assay. I think we also have in vitro biological assay, but depend on what is your aim, what is your objective of your work. If, for example, your work, the conclusion or the application will be linked into the human health. Now, might be you need biological assay. There are many in vitro biological assay, inhibition of hemoglobin or LDL oxidation. For that method, you need time consuming because you need to isolate, the LDL from plasma, and then isolated LDL, because you need a train, you have to train yourself because it will take some time. Sometimes also the result, of course, when you're dealing with the antioxidant capacity, the assay, the method, especially the in vitro biological assay, the precision, I think very hard to get, the good one, because a lot of factors, could influence your result. And then I think the famous, I think now, in Malaysia, CAA, not CIA, CAA, cellular antioxidant assay, based on the assay base. I think in FRIM, I think they have service for the CAA. They have, I think, the method for the cellular antioxidant assay. And then I believe there are also university develop this CAA. And then this CAA will be very good because you are working with the biological on the same time, you can see also the effect of the bioavailability of that particular compound into the cell. Okay, I think there's a bit our case study of antioxidant. Okay, we're working with one of the fruit from Sarawak. I think Dabai. I think maybe you are not very familiar. Some of you very know. Okay, because this food very good, yeah, commonly consumed, yeah, during toward the end of the year in Sarawak, yeah, among the Iban people, because that time we are very interested, to analyze or to explore the underutilized and neglected, yeah,
Hello?
food and yeah. Hello. Okay, give me about 1 or 2 minutes, yeah. Hello.
Please proceed, Prof.
Okay. Okay, continue. Sorry. All right. So because here we started, our work with using the in vitro assay first, yeah, based on the FRAP or the PPAs, beta carotene, bleaching assay, and then we found out it's very good result, and then we proceed. We are using the animal model to test, whether this food, yeah, can have some effect.
Hello, assalamu alaikum.
Yeah. Hello. Okay.
Hello, assalamu alaikum. I'm
Can I
hello.
can I finish my presentation first? Okay, and then from there we found
No,
out
no.
also very good, result, yeah, on the in vivo, and then that's why you cannot stop, at that point you have to explore, in this extract what is the compound, could contribute or might have some idea. You have some idea what the compound, so you have to use the analytical method to determine or to identify what is the compound could act as the antioxidant, because you need an instrument, a method, and then also you have to proceed, for example, when you want to have the effect on the human, because you have to carry out the human work, and then you on the same time you can analyze the plasma total antioxidant status. From the result you can compare whether is it your result, in line with support, by the animal work or in vitro.
Hello.
Go very detailed, yeah. For example, maybe you would like to see the mechanism. You have to study using the molecular level, look at what is the mechanism, From there you can have a very comprehensive understanding, the whole picture about your
Hello,
sample.
assalamu alaikum.
And then should we ban, because there is one short communication,
Hello, assalamu alaikum.
published by our group, in this food chemistry, I think my team, I think like macam satu bas lah, ramai. So we publish, the antioxidant activity, should we ban, the in vitro method. Okay, and then from the our short communication, because we concluded, that important of antioxidant capacity or activity, in vitro, chemical method, when you combine with the instrument method like a liquid chromatography, and then at least, some of the in vitro, biological tests also you have to support with some of the in vitro biological tests, for example cell line, or simulated, digestion or in vivo assessment. And then I think very important, antioxidant, metrology, you have to study on the method development and validation. If you would like, to maybe you want to offer for service, to the industry, you have to study on the repeatability, reproducibility, whether the proficient, test, of that one very important to have the reliable and the accurate result. Okay, I think we have, I think not quite recent, I think about 3 or 4 years back, we write a book, Antioxidant Assay, Principle, Method and Analysis. This one a very simple, very simple, very simple process. I think very easy, to follow for the student. We we target to the undergraduate student, postgraduate student, because that one we also have a very simple introduction, principle of that particular method, and then we have the procedure, and then we have some the troubleshooting on that, and then frequently encountered the shortcoming during the analysis and troubleshooting. Because if you're interested, because I have still many copy in my room, because that's why
Hello, Assalamualaikum.
that one I think very good, for if I think very good, for you if you are interested. I think maybe we can have a very discount, price for you to finish all the antioxidant essay, because I would think, because maybe in the 2 or 3 years, because this book, not any more interested by many student. And then my concluding remark, requirement of the analysis, of all aspect, for example, you have to have the analysis, of the chemistry mechanism, for example, the reaction, in various test system, carefully and accurate quantitation of all reactant and product involved, and then the importance of in vitro method or assay to determine antioxidant capacity or activity in food samples is still relevant. Only one in vitro assay is not acceptable to make a sound conclusion. Therefore, analytical method like HPLC, LCMS, UPLC, and others, and in vivo experiment should be involved. Method development and validation of antioxidant capacity or activity assay should be done for reliable and accurate parameters. Thank you. Thank you so much. Thank you, Dr. Zaibunnisa.
Okay, thank you so much, Prof. Amin, for that very concise and very informative information on the methods, antioxidant capacity, determination of antioxidant capacity in food samples. Actually, we have a lot of questions in the chat box. They are actually interested, participants are interested for the slides and also the recorded webinar for today. Later we will get the permission from Professor Amin if it's we would like to inform if it's available, the soft copy of the PowerPoint and also the information about the books shown also will be shared later. So now is the Q&A session. I noticed just now there are some participants who really wanted to ask question, Prof. Amin, from just now.
Yeah.
I noticed that. So now we can actually open the floor for Q&A session. If there is any question, please you can write on the chat box or you can actually ask Prof. Amin directly. Please, any question from the floor?
I have. Hello, Assalamualaikum.
Waalaikumsalam. Safa, yeah, Safa.
Hello,
Yes.
Assalamualaikum.
Welcome, Doctor.
I am calling from Bangladesh.
Great presentation. Can I ask you some questions?
Okay.
Okay. The first one, are many
Yes.
ways to determine the antioxidant activity in food samples. So it is necessary to determine all these methods food samples, or should we be satisfied with one method, and which one is better and recommended to determine?
Okay, thank you, Safa, for your question. Okay, if I'm, understand you clearly, you're asking about which method, we have to choose, to analyze your antioxidant capacity, whether is it one enough, Okay, because I think also very difficult, to answer you because okay, of course, one method are not enough. One method not enough. But you have to look at what is your objective of your work. Whether you want to screen your food sample or what you want to do with your sample. Maybe you want to relate maybe for the lipid oxidation as antioxidant agent for the lipid oxidation. Maybe you have to choose something, the method where the mechanism or the principle tally with your objective.
Yeah.
For example, maybe you would like to study on your food sample. You want to relate with the heart disease.
Yeah.
Will be dealing the lipid oxidation. Maybe you have to find the method where mimicking, the mechanism, the one you are interested. Okay. Yeah.
Okay, thank you, Doctor. And I have another one question, please. About your book, Antioxidant Assay, is available in internet, or how can I got this book? How can I got?
Safa, you are from where? From Malaysia or from?
Yes, I am here in Malaysia now.
Oh, okay. You can email me. You can get my email from. You can get my email from the organizer, and then maybe you can email me directly. I think
Okay.
I will. Yeah, I will reply you.
Thank you,
Okay.
Doctor. Thank you so much. Assalamu alaikum.
Okay, there's one question from the chat box, Prof. Amin. Is it necessary to make correlation?
Again.
Is it necessary to make correlation between antioxidant activity, D2PH, and level of total phenolic, total flavonoids, and vitamin C? Do you need to correlate?
Okay. Again, I think thank you for the question. I think again, you have to refer what is your objective actually. If you want to look at the contribution of your compound that you are interested, to the antioxidant should be okay. But you have to see also the number of your sample. If your number very little, so I think the correlation not very good. You have to have many sample, and then your correlation will be very strong. If you are only sample 1 or 2, I think not very good correlation, because correlation normally you have to have more than, for example, 20 or 30, because you have to come up a very strong correlation. And then again, you have to ask. I realize that many article or many study, they try to correlate between the antioxidant and the antioxidant assay, the method and the antioxidant compound. I think will be very good, no problem. And then on the same time, they also make a correlation. They make a correlation between the method and method. I think for me, what for,
From Bangladesh.
because
Please listen me. Hello.
yeah. Okay.
Hello.
Yeah.
Yes, Miss Salim, you can ask Prof. Amin your question.
Miss Salim from where? Because I think for the chat box, I'm not going for the chat box. I rely to the Zaibunnisa, to read, because I think many, I'm not sure.
Okay.
Excuse me, sir.
Yes.
Sir, I am from Pakistan, and I joined sometime later that class.
Yeah.
Yeah, I want to know, sir, that please, I found this class in any link, etc., or recorded audio.
You can Mr. Shah, if you are registered as a participant, we will email to you the information later. Thank you.
So, thank you. Ji.
Okay, Prof. Amin, I'll just read out some more question for you.
Okay, good. Yeah. Okay, no
Okay,
problem.
Okay, may I know what is the mechanism principle involved in primary, secondary antioxidants?
Okay, all right. Because that one you are talking about the primary and secondary antioxidant because it's the compound, right? Okay, I think the same, the same principle because the mechanism based on the in the slide I think I have been cover several mechanism of these antioxidant, regardless whether is it the primary or secondary antioxidant. I think the mechanism more or less I think the same, but you have to understand that not all compound have the same mechanism. Some may have different because I think the compound also they are very smart, because they are according to the system, the food system. Sometimes when using this different system, they have act differently, because antioxidant sometimes they can be antioxidant, sometimes they can be pro-oxidant. That's why you have to take into consideration. I also have some, I think, the question from the student, if I'm not mistaken, about the optimum, what is the optimum concentration for antioxidant that useful to maintain food product, shelf life. But we cannot give a correct optimum concentration of the antioxidant. You have to make some study. You have to test at a different concentration because different antioxidant, maybe they have the different activity, depend on the food, depend on the food product you develop, because not all will be the same. And then the maximum concentration that safe to consume, again, you have to make a study on that. You have to test. You need to use toxicological assessment to test. Using this, look at, you have to plot the IC50 of that. And then are there any surveillance activities by authorities? I have no idea. I think there is, maybe under the national pharmaceutical bureau, because this is antioxidant, normally we dealing with the food under the bahagian kawalan kualiti. If under the supplement will be under the pharmacy. And then which method has a higher sensitivity? Because again, this one I cannot tell you. I have no idea because depend, because now there are many methods, many assays, because you have to test on which one they work, for your system. And then sample preparation for each method is different. I think the sample preparation more or less quite similar, because you extract with water or organic solvent. And then which one of the method is the cost effective? Because I have no idea. I did not doing on this calculating the cost, but I think if you can ask the company or the industry who offer service, you might get the answer. And then how long duration procedure? Okay, that one depend from method to method, the duration to get the result. Sometimes very fast, sometimes will take almost 24 hours. And then the constraint for mission researcher, yes, yeah, I think funding, yeah, of course, funding, yeah, and instrumentation, yeah, because that one, the two factors, yeah, I think will hinder, yeah, us, yeah, from move, yeah, in this antioxidant research. Okay, I think one method not enough. Yeah, previously yes, nowadays more than that. Not only more than one method. You have to support, yeah, or you have to add the instrument method, yeah, HPLC, and then also you have to use in vivo, yeah, animal, yeah, or human, yeah, work on that. And then, is there any rapid kit? Yes. Yeah, in the, I think in company, yeah, I think they have developed, yeah, come up with this rapid, yeah, kit test, yeah, if I'm not mistaken, or like DPPH, yeah, and others antioxidant assay, but can be used at home, yeah. I think, but you again, you have to ask yourself why you want to use, at home. Is it for, for the consumer health or something? That one you have to ask, whether is it necessary, yeah, to conduct at home. Okay, that one I cover, yeah, the question from the student, yeah, I think, and Dr. Zaibunnisa.
Okay, Prof. There are some more questions in the chat. I'll pick. There's so many questions actually, Prof. they, want, they want to
But
know about. It's a very interesting topic actually.
Yes.
Okay.
that's why Dr. Zaibunnisa, I also, when you ask me to share, I also don't know which one I have to focus because I have so many things, to share.
Yeah, yeah.
Yeah.
Okay. This one, Prof. One question. The question is actually, what is the nutritional effect? I think what they're trying to ask is, what is the difference between natural antioxidant and synthetic antioxidants if you consume?
Okay, I think the natural, of course, I think you all know, yeah, that mean extracted from the food sources, yeah, with the plant or animal, yeah, or marine, yeah, a marine source. But the synthetic normally from, for example, they are synthesized, yeah, in the, for example, they have the structure, yeah, based on the natural source, and then they know the chemical structure. So they synthesize by their own using this the chemical reaction. They come up this the antioxidant. This called the synthetic. Yeah, because BHT, BHA, TBHQ. Yeah, that one they are based on the petroleum. Yeah, many antioxidant they are based on the petroleum source, based on the petroleum, not from the food. Of course, because depend on the dose also, because one of my student, my PhD student, did work, to test different natural antioxidant versus the synthetic antioxidant. Okay, on animal model, but at certain dose should be okay. If because if you limit the dose, they might affect the liver function. Yeah? In terms of the histology, you can look at there is some toxicity effect of this the dose, yeah, of this the synthetic the antioxidant.
Okay. That indicates that natural is always the best, Prof, compared to
Yes,
synthetic.
yes.
Okay. Okay. There's one question, Prof. What is the significance of antioxidant? What is the significance of antioxidant capacity studies across the field, such as biochemistry, molecular biology, and zoology? Is it necessary to do antioxidant capacity study?
Yeah, I think the antioxidant capacity, I think started, yeah, with the chemistry field, yeah, in chemistry field, yeah, natural product, yeah, in the food industry, yeah, food science, yeah, food technology, and then now there are, I think not now, I think last 5 years, yeah, I think nutritionists, yeah, and then also pharmacologists, yeah, and then molecular biologists. they are really want to use this essay in their work as a screening part. Because before they proceed further, they want to have the screening, or they would like to have some basic information about the antioxidant before they proceed very detailed in their field. Because for me, necessary, very still relevant to look at a first glance about the sample or the food sample you are going to work further, because have some idea. If you want to work with any food sample, first, if you would like to know more about this one, first maybe you have to start with a very simple essay to have some idea. From there, you can proceed further. I think this is very important. Otherwise, you have no idea. You cannot just simply, jump, to human study of that. Although you don't know whether is it this food sample, has antioxidant, activity.
Okay, another question, Prof. What is the most acceptable way to express the antioxidant capacity result in order to publish in high impact factor journal?
Okay, I think, yeah. Okay, all right. So, okay, I think, publication, one of the, I think, concern, by the researcher, because, I think the student also, not only the researcher, the student also, the expression of a result, based on the antioxidant capacity, the essay, you have to look at, how, most of the scientists, in the world, expressing. Because, for example, I look at DPPH, I give one example, DPPH. Previously, we, because my group, express as percentage, percent of scavenging activity. Previously, I think no problem, most of the people, the journal, the journal, they are accept, they accepted that kind of how you express the parameters of antioxidant capacity based on the DPPH. Nowadays, not nowadays, I think last 5 years, they are moving to express as IC50. Because IC50 will be very good, because they are not like, IC50, but you have to have a different concentration, and then to get the IC50, the 50%, and then from there, you can have when you have this IC50 lower, the capacity will be higher, the capacity, because the lower your IC50. I think depend on the antioxidant capacity assay you use. But make sure you have to express, you have to look at what is the currently, the current trend, how they express, because you cannot simply, based on the previous information, you have to update, yourself. Because I cannot tell you, I cannot tell you, okay, this antioxidant activity, how to express. Normally, the expression, you have to have, the update, on that, how, the people express. I think like, the PPH because the PPH previously percentage, now they are moving to IC50. But again, you can have some, your innovative, or creative way of expression of antioxidant activity, no problem. As long as you understand what you express and then you are able to explain, should be okay. I think, the journal, they're also looking forward, to have I think the reviewer not the journal the reviewer, they are looking forward when you can come up something how to express, because they like, something they call novelty, a new thing, about the expression of antioxidant capacity.
Okay, there's one interesting question in the chat box, Prof. How can we explain that certain compounds or extract having antioxidant activity at low concentration loses their activity at high concentration?
Okay, I think this one quite straightforward, because if you would like to see the low concentration, of your compound you are interested, and then also versus the high concentration, you have to run at a vary of the concentration. When you vary the concentration, you can see normally, not necessarily, the higher the better. Meaning that, the high concentration, sometimes your high concentration of antioxidant not necessarily will give the high antioxidant capacity of that particular compound. I think the optimum will be very important here. Because that's why, again, I said, antioxidant, at certain level, they will be a pro-oxidant. So, at optimum, a concentration, will be necessary for you, to work on. That's why, you have to also consider how much your compound absorb, into your body. Is it at that concentration or is it that a low concentration? Because in our plasma, a very minute, amount of the antioxidant compound. Sometimes also the compound, they have a different form, not the one, in our food. For example, in our food, normally like polyphenol, there are many glycoside form. Because the one absorbed into our body will be the glycon without sugar moiety. That's why, for example, when you test phenolic, glycoside form, the one, in your plasma will be the glycon form. That's why you have to have some idea, before we come up something conclusion. You have to understand about the bioavailability, a lot of things. That's why the method, to determine antioxidant capacity. Also, antioxidant, there are many things, can be shared. I think, we cannot share, in this only one platform because there are a lot of, debate here and there. That's why, based on my experience, more than 20 years, dah tua ni, Zaibunnisa, So I, talking to many the prominent scientists in the world. There are many, there is a lot of things, debate here and there, until now about the antioxidant capacity, the antioxidant, a lot of things. Because make sure, because all those not don't stop you from proceed in antioxidant. Don't, after this webinar, some of you maybe want to change. Oh, no want to work with antioxidant because not very good. No, because because for me, because we have to have passionate, in our research work, in our whatever we involve, because will be more new thing, you will get. The challenge, I think, will be very good. Yeah.
Okay, Prof. One more question here. I think the question is trying to ask if antioxidant extract been added in a complex food system, how do you actually measure the addition of the extract, the capacity of antioxidant? Maybe the food itself already have some antioxidant activity, capacity itself. So how you want to, how to improve the method, one, and then how to reduce the error?
Okay, all right. Okay, I think this one for the added of the antioxidant in the extract, or in the food system. That's why you have to have, like, first, if you're working with the fresh sample like fruit or vegetable, you extract this fruit extract or vegetable extract first. Maybe you have to have this. You have to test the antioxidant capacity, and then when you add, the additional antioxidant, you have to test again. But when you have the processed food, from the market, because the process, they call already the antioxidant, compound already in this natural form, and then they add the new thing. That's why, very hard. You cannot. Very hard to differentiate, because when you're working with a processed food, when you're working with something, the fresh, fruit or vegetable, very easy. If you're working with the processed food, like I think you can name it, many processed food, now they have the added, with all kind of antioxidant, because again, some of the compound, although they are not considered antioxidant, they can have the mechanism as an antioxidant. That's why very hard. That's why if based on the assay, based on the clinical assay, but we need to have like using instrument HPLC or LCMS to check what compound, for example, could, contribute. We have some idea, for example, what compound you target. Maybe phenolic or maybe other compound, other non-phenolic also. That's why you need to have not only assay, in vitro assay, but you need to have the instrument like HPLC or LCMS. Nowadays, people go for LCMS, GCMS, they go very detailed. A lot of things now. They are, I think now the sign, they are very dynamic. Sign very dynamic. Antioxidant capacity assay also very dynamic. They are not static. If we are not follow, for a year, so we will miss many things about the development in antioxidant. I'm also sometimes miss because I'm not follow because here I have to focus other thing because now when I go Google and I find search from the journal I think now they are progressing very very fast in the antioxidant area work. Tak tahu lah jawab ke tidak.
Jawab, Prof. Okay, ada lagi question ni, some more question, Prof. Which in vitro antioxidant method is most effective in explaining in vivo antioxidant activity?
Yeah, okay. I think in vitro result can be used to have some idea before you proceed using the in vivo test, in vivo study. First, normally you have to have some idea on the in vitro before you proceed with the in vivo. But not all. Some of the, I think I know some of the people, although they have I think the low antioxidant activity or capacity of that particular food, they would like to also to proceed using this in vivo using the animal model. But suddenly they found I think the good result. That's why sometimes you cannot just simply before you proceed to the in vivo work. Because for me, this is a systematic process. In the systematic process or in the in the thesis, normally you have to follow for example step by step process. For example, you work with in vitro first, then you have some idea, maybe you have some few method of the in vitro assay. From there, you can decide. For example, which sample you want to proceed using the in vivo study. Based on 5 sample, maybe you can downgrade into certain sample. Maybe you can, smaller your sample size. Instead of 5, maybe you can focus 2. Because some of the people, they are screened for 10 sample, and then they want to choose only 2 sample. And then a lot of things you have to do. You have to vary extraction medium, temperature, and then extraction time, and then method, a lot of things. But you have to have some idea what you are aim for that particular sample, what you want to focus. You want to go for look at anti-diabetic properties or lipid, inhibition of lipid, or cancer, or other thing, or obesity. There are a lot of the essay. You have to do some homework before you choose which one I think the most good for you, the most work for you, the good very relevant for you. You cannot simply follow, my lab already established that particular assay, I can use. No. Then one you have to ask yourself whether the mechanism, is it follow, for the next process of the analysis. This one very important. You have to do a systematic, but not necessarily, after you got low antioxidant capacity using in
Hello,
vitro,
Assalamualaikum.
you cannot proceed with the
Hello.
in vivo. But that one, you if you have some, instinct, oh, maybe this one maybe work on the in vivo. I think, as a scientist, no problem, you can go. I think you can test. I think that one, no harm, to have some idea on that.
Miss Celine, do you want to ask question? If no question, I'll proceed, Prophet.
Okay, yeah.
Yeah, there are some samples that have color compounds. Will color compounds interfere with antioxidant determination?
Okay, yes. I think yes, but depend.
Hello, Assalamualaikum.
Yes, but depend on
Hello.
the concentration you use. If you dilute, if you diluted the
Hello,
sample,
Assalamualaikum.
yes, if you dilute, if you dilute the concentration of the sample might not, influence your result, because most of the in vitro assay based on the spectroscopy, spectrophotometric method based on the color formation. If, for example, you have a high concentration of your sample, might influence the result. The reagent you use might influence, because that's why you can see the high absorbance when using the DPPH, right? If you have a colored sample, you have the high absorbance, more than one. That one not very good, more than one. That's why you have to dilute your sample first. Because from there, you can calculate back, you can just calculate back how much you dilute and then to get the result. Because this is very important, you have some idea. That's why working with this antioxidant assay, you have to be patient, you have to be, sabar-sabar sikit, because not very easy, sometimes they will test, your kesabaran.
Okay, Prof, there are questions coming in.
Okay.
Is it okay for you, Prof? Okay, all right. Okay, there are questions saying that how do you tell when you determine antioxidant capacity in food? You can consider the food as a high antioxidant capacity food. I mean, what they are trying to ask is, what is the amount can be considered as a high antioxidant capacity?
Okay, the one thing for to give to say this your sample, your food sample have high antioxidant capacity, you need to compare with the standard. For example, you need to compare with what? Depend on the antioxidant assay you use. For example, you are using DPPH. What the normally DPPH? What the mechanism of DPPH? What is the best standard? The antioxidant you have to use. Whether you want to use vitamin C, or you want to use vitamin E or BHT. For example, maybe your sample, you want to test on the phenolics compound. Maybe you want to use quercetin or any the isolated compound. You want to compare your sample and the standard, because the pure compound. From there, you have some idea whether, because you cannot compare, between your sample with other sample published, because the condition might be different. The condition, the time, the pH, but you need to compare with your standard. What standard you want to use? Maybe you are the pure standard, or maybe you want to use something which is already established. For example, I think you know about the standard reference material, a certified reference material. Maybe I have no idea whether they have this standard reference material for the antioxidant test, because the one you need to ask the supplier. If they have, will be very good. If they have the certified reference material, will be very good for you, because you can run this certified reference material and then you can compare with your sample. From there, you can decide, whether high or no. Because otherwise, very hard, to make a decision, because what I said previously, they have different mechanisms, and then also complexity, because also depend on the pH, the solvent you use, also the other compound, present in your food sample.
Okay, Prof, there's one more technical question on the method itself. The problem faced by this participant is if the sample turned the DPPH region from purple to brown. They said that DPPH is not a suitable method for that particular sample. What is your comment on that, Prof?
Okay, your DPPH, you working with the DPPH, and then your sample from purple turn to brown, is it? Turn to brown, because I have no idea what type of sample you use. Because some sample, the exact exact sample, sometimes your exact sample, when you expose into the, into the oxygen, or to the light, they might already oxidize. This is not they already oxidize before they react with your DPPH, the radical, the stable radical cation. And then again, you have to check. That's why I think the quality control very important. You have to check in terms of the chemical you use, whether is it the expired, or maybe kept, not in the proper condition. There are many things. That's why, when you want to work with DPPH, better you work with your standard first. Working with the standard first. If you're working with the standard, has no problem, then you're working with your extract, has problem, might be, the compound or any compound present in your extract, because we have no idea. Maybe sugar. If you're working with the high sugars, high carbohydrate sample, or maybe a protein sample, they might, influence your result. Because I have no idea. Because that's why when you ask me about this shortcoming, I think very hard, Otherwise, I can discuss with you, this, that. Because previously, there are many things, there are many factors, could influence your result. And then whether you are DPPH, suitable or not for you. Again, maybe, for example, what I said, DPPH, you cannot use only one DPPH. You have to have more. At least you have 3. Maybe you can test other assay first, and then from there you can see whether which result more consistent, more reproducible, for your sample.
Okay, thank you, Prof. Okay, this is one question from one participant as well. Concentration of antioxidant is strongly depend on the standard curve constructed by the researcher. Let's say the absorbance of sample within the range 0.1 to 0.3, and the standard curve, do we have to make the standard curve from 0 to 0.9, or is okay we just plot the standard curve within 0 to 0.4 or 0.5?
Okay, I think Okay, I think if, for example, your sample, 0.3 to 0.9, I don't know, okay, 0.3 to 0.9.
0.3 to 0.9, yeah.
Okay, 0.3 to 0.9. The one you with your sample, right? And then the standard, because for example, your standard suppose, because your sample suppose within the standard. Meaning that you have to have more than that, your standard. Maybe 0.1 till, maybe 1, something like that. Because if, for example, very hard, because I think standard also very expensive, because if you prepare many concentration, might finish all your standard. Maybe you can dilute your sample first. You dilute your sample, make it the absorbent lesser, and then from there you can plot. You can plot your standard, more than that, because your samples are put within the standard. Because you cannot have your sample maybe out or below the standard, because not very good in terms to get the calculation of the concentration. Although they can be done by using the y equal to mx plus c, but I think practically your sample, the concentration of your sample, the absorbent of your sample within the standard.
Um, okay, Prof. This is another question. How many methods do we need to carry out to conclude sample is having high antioxidant capacity?
Again, I look at my handphone. That's why. Okay. Again,
Sorry. How
it's
many
okay.
to conclude your samples having high antioxidant activity? At least minimum how many methods that we have to carry out?
Okay, for me, you have to have more than one method. For me, I think it will be very good if you have a 3 method or 2 method. Yeah, different mechanism. Different mechanism, maybe from the set, single electron transfer. Another one maybe from hydrogen atom transfer. Not enough for the in vitro, not enough for the in vitro chemical assay. You have to have using instrument, HPLC or other machine, to have some idea what is the compound. You, for publication purposes, nowadays they are not accept, they are not accept any article or any work, any manuscript, only one or two. If you have two, okay, maybe you can say, oh, one not enough, maybe you have two. Okay, two should be okay. Maybe one from single electron transfer, another one from hydrogen atom transfer. They need more. They want to have what is the compound could contribute, to this antioxidant. So they need you have a identification or quantification of that particular compound. And then you claim, oh, this for the anti-diabetic properties. They need you to run the animal study. What effect of this compound to the animal study? Why this one very important? In your discussion, you are able to discuss this compound could, might contribute to this effect based on your explanation. And then also you support with animal study. That one, your work will very good. Quite, can be accepted, because you have idea. For me, that one very good because they are not, looking for the mechanism. Because nowadays, when you, I believe, all of you have update, your any research, or any publication article, published in journal, recently. You can see many articles from where, from China, you can see from China. How they are, when you read, the article, you can see how their work, not only very superficial, they are very detailed. So, if your paper, you want to submit for publication at an international journal, and then the publisher also receive article from other country as well, from China. So they are trying to compare, because our analysis, very simple. And then compared to the China, scientists, they have this work, and then they have very detailed, on the molecular everything. Sometimes, when you read, you don't think, oh, they are able, to do it, until that point, because you can see how they are developed, in this, antioxidant work. That's why for me, beside the antioxidant assay, based on the chemical assay in vitro, you have to support with the analytical instrument, and then also with the in vivo analysis. Of course, you say, oh, how long will it take for you to do? Of course, if you have a very good collaboration, or very good, have some idea collaboration with your supervisor or with your co-supervisor. That's why the time for collaborate, not only within your department, within your faculty, also not within your university. Nowadays you have to go beyond that. Others university, not only in Malaysia, in overseas. Now I think it's the future, the collaboration now. I think not we cannot work alone nowadays, because we have to work borderless now. Now borderless, we have to work. Otherwise, you cannot depend our facilities at our lab, because we have to collaborate. Otherwise, you are not moving very fast.
I really agree with you for that, Prof. Amin. Okay, so I'm really sorry. There's so many questions coming in. Again, more coming. Are you okay, Prof? To answer? Okay.
Okay.
Okay. There's one question here. Based on the mechanism, is there any recommendation? Sorry. Using IC50, is there any value that we can calculate to conclude the sample is having good antioxidant capacity?
Okay, I see 50, the unit based on the microgram, right? Because when you have the IC50, you have to run the different the concentration, this mean milligram versus with this one, the IC50, for example, this absorbent, and then for there you are able to get the unit. Normally your unit will be in the milligram per mil or something. If your IC50 lower, 0.05, and then you compare with 0.5. The 0.05 will be better, the scavenging activity of that particular food compared to the 0.5.
Okay. Okay, Prof. Are there any assay kit for antioxidant detection?
Yes. I saw in the internet because I have no idea whether in Malaysia we have a company offer this product at the kit. I think they have now. If I'm not mistaken, DPPH, and then others ST as well they have. Because again, maybe you want to have a comparison, but you can compare, you can buy, and you can compare the one you prepare, based on the preparation of chemical, in your lab. You can make a comparison, because I never test any kit, antioxidant kit in the market.
Example of analysis carried out to determine the enzymatic and non-enzymatic antioxidant for total antioxidant.
Because I think they have like xanthine oxidase, because xanthine oxidase, one of the okay, I have some here. Okay, if you look at there is the in vitro enzymatic assay, like SOD activity assay, because you based on the you assay the activity of that particular enzyme. For example, SOD, and then glutathione, ascorbate, peroxidase, and then xanthine oxidase, because when you working with, I think, xanthine oxidase, most of the people working with a cosmetic, they would like, to have this, they're using this assay to check, whether their extract, of their food sample has a very good in this xanthine oxidase. And then peroxidase activity, catalase activity, for example, you want to have this one of the assay based on the enzymatic assay.
is there any maximum concentration of antioxidant that we can consume? Will it cause toxic to body?
Depend. It depend. Yeah, depend from one antioxidant to another antioxidant. Again, you have to have a study. Yeah, you need to carry out study. Maybe you can use first using the cell culture to look at the IC50, yeah, the toxicity limit, and then if you really want to apply to the human, maybe you have to check, this using the animal, and then from there you can have some idea, because one antioxidant, different, between one antioxidant to another, they are not the same. Also based on our system also, sometimes maybe for me I'm okay at that regular concentration, but for maybe for Dr. Zaibunnisa, that concentration will be toxic, for her. That one depends on the system, biological system as well.
Okay, few more questions, Prof. EC50 and IC50. IC50, is there any difference?
Okay, I think I have been, I also, see some of the report, they put it EC50, sometimes IC50, but I think if I'm not mistaken, how they are calculated, the result might be similar, because I think EC or something, because I think will be similar for the DPPH. I don't know for other assay, but for the DPPH, some of the researcher, their report as IC50, commonly IC50. There are some EC50. I think will be the same. Will be the same.
Okay, it can is it possible for compound to give false positive antioxidant capacity results?
Yes. They have they call the pro-oxidant. They have the pro-oxidant, because we have okay, that one based on our work, because we have the extract of this cocoa extract. We have a cocoa extract, high concentrated of this polyphenol, and then we give to the animal. This animal, we have the diabetic animal, and then we just force feed, this animal. And then if we force feed the animal at certain concentration, all the animal will die, because due to the toxicity of that particular compound. When we give to this the normal one, okay, but at the disease state, you cannot have a high dose of this. They consider semi-purified the antioxidant extract. Also the extract also very harmful for the disease state of this animal. That's why I think will be applicable for human as well. If you have a disease, you have a diabetic or other non-communicable disease, you have to make sure, in terms of the consumption of this, antioxidant, supplement, for me supplement or extract, but for maybe as a vegetable or ulam or salad should be okay. But if a concentrated form, might be give some harmful for you.
Okay, I think that's the end of our session for today in Q&A session. Very helpful. Okay, that's the last message in the chat box. A lot of vast knowledge and a lot of information we gained today. Thank you very much, Prof. Amin, for joining us in UiTM, and also everyone all over the world that watching the webinar today. So with
I'm very happy, to have this one. I would say this the Santu, Santu, Dr. Zaibunnisa, Santai Satu, Santai Satu, every Santu. I think will be very good if we have Santu, to this one. I'm very happy, to share whatever I have. I'm not, know everything, because this is based on my knowledge, my sharing, because I think the participants sometimes they are knowledgeable, in terms of the antioxidant.
Yes, Prof. Just some information. This webinar actually organized by student itself, our master student coursework. So thank you very much for the student also. The program manager is actually Miss Sharifah Nuruljannah Syed Imran and her team for organizing this sharing session. Um, with that, I would like to pass back the session to the MC for our concluding session. Thank you very much. Assalamualaikum.
Thank you.
Thank you to our moderator and invited speaker for a very interesting and informative session just now. Now, I would like to invite all the participants, including the moderator and our invited speaker, to open the video for our group photo session. Okay, guys, let's open the video so that we can see all of your beautiful and handsome faces. All right, and please wait for a few seconds because we have to screenshot a lot of pages in order to capture all of the participants today. And please smile for the camera. Right. Okay.
Thank you, dear. Thank you. Thank you so much for informative information to sharing us.
Thank you, guys. And then I would also like to remind everyone to fill in the feedback form, which we already provided the link and the QR code in the chat box. Thank you for all the feedback.
Thank you so much.
Lastly, for the e-certificate, we will issue the e-certificate within 3 days maximum, inshallah. I think that's all the time that we had today. Thank you for joining us for this webinar. We actually have another webinar next Saturday on the 19th of December. The topic will be on nuclear magnetic resonance, NMR. Yes, there you go. That's the topic for next Saturday webinar, nuclear magnetic resonance in food analysis. I'll see you guys. Hopefully you guys can join us for this webinar series. The invited speaker is from Thailand. So this will be internationally invited from the Thailand. In the meantime, stay safe and follow the SOP. Thank you all so very much. Wassalamualaikum warahmatullahi ta'ala wabarakatuh. I'll see you guys next week. Thank you.
Thank you. Thank you. Thank you, Ikrami.
Thank you.
Thank you,
Thank you.
Dr. Nabisa.
Thank you,
all.
Prof.
Thank you.
Okay. Bye-bye. See you.
Bye-bye. Thank you, Prof.
Prof,
Alright.
thank
Thank
you.
you, Prof.
Thank you,
Alright.
Prof. Thank you so much.
Bye.
Assalamualaikum.
Thank you, Prof.
Most regard. So much appreciate you, Prof.
Thank you, Prof. Amin. Thank you, Prof.
Please don't forget to send the PPT. We are so much interested in the PPT of the presented by the moderator.
Ugh, how to get out?
That was awesome for everyone, I think. And I wish that all of the participants will join next week in this seminar. So please inform all of them.